Hello, in this video we're going to be looking at the beginning of the digestive system. This section of PowerPoints focuses specifically on the anatomy of digestive organs and groups them into functional classes. We're going to start in the first video by looking at an introduction to digestive system anatomy. So to begin with, most of the organs we consider are part of the alimentary canal, essentially just the hollow tube that transports food and waste from the mouth to the anus. Accessory organs exist outside of the alimentary canal, and they help with processes of these alimentary canal organs. Great examples of accessory organs would be the salivary glands in the mouth, they'd be the pancreas, the liver, the gallbladder, so forth and so on. It's really important that as you go through each of these organs that you're able to classify them as either alimentary canal or an accessory organ. All the alimentary canal organs, because they are a hollow tube, have walls that are formed of more than one layer. We also refer to these layers as tunics. Now, the tunics are something that you would have looked at in lab, and there are four major tunics: the mucosa, the submucosa, the muscularis, also known as the muscularis externa, and the outermost is either classified as serosa or visceral peritoneum. Or adventitia, depending on where you're looking at the organ. So to begin with here, the mucosa itself is the innermost lining. It does make contact with digested or ingested food. And this hollow space inside of the mucosa is known as the lumen. And so this is showing you right over here the mucosa, the innermost layer, right through there. And there's the lumen. Now, the part of the mucosa that actually comes in contact with the food itself is going to be made up of epithelial tissue. It's important to pay attention to what kind of epithelium forms the mucosa of individual organs because this is going to denote function. So, for example, in organs where you might see more abrasion, either at the beginning of digestion or at the end with fecal material, you're going to want a thicker epithelial membrane, something like stratified squamous epithelium. In the organs where you want to see a lot of secretion or absorption and you don't need that thicker lining, then you're more likely to see simple columnar epithelium. So pay attention to this as you go through these individual organs. Now there is a thin, smooth muscle layer here known as the muscularis mucosa, and this is not going to be responsible for propelling food through the organ. Rather, what this does is as it changes shape, it actually can change whether or not this is a rippled or a flattened surface that food is coming in contact with. That's important because it can change how well this epithelial lining is going to be absorbing or secreting based on the surface area available. Now, just outside of this or superficial to this is the submucosa. The submucosa contains a number of things, including blood vessels, lymphatic capillaries, secretory glands, and other lymphatic tissues, such as Peyer's patches that we've seen. And it contains a nerve net known as the submucosal nerve plexus. And so you can see that right through here. Here's that submucosa. You can see some of those glands. Here you can see some of the yellow nerves, green lymphatic vessels, and the red and the blue circulatory vessels. As you'll notice here, the glands that you see in the submucosa, if they are exocrine, which most of them are, open with ducts here to the open surface of the epithelium of the mucosa. And so, for example, if these are secreting digestive enzymes, then those digestive enzymes would travel from the gland down a duct and then coat the inside of this mucosa and the food that's inside the lumen. Then you move to the muscularis externa. This is between 2 and 3 smooth muscle layers, and they're used for propulsion and mechanical digestion. Typically, you'll have an inner circular layer and an outer longitudinal layer. At thickened regions, the muscularis forms sphincters, which regulate entry or exit of food from a particular organ. And this is going to be controlled by a myenteric nerve plexus. Myo means muscle. And so you can see that here. Here's that nice thick muscularis externa, the inner circular layer. Here's that outer longitudinal layer running up and down the length of the organ. And here's that myenteric plexus that runs to all the muscles there. And so this is the nerve net and the muscle that's actually regulating movement and digestion through an organ itself. And of course, finally, you would see either your serosa, which is most frequently known as the visceral peritoneum, or you'd be looking at adventitia. When you take a look at mesenteries, mesenteries are a great example of what this outermost layer of a tunic could be. And so, for example, if you happen to be looking at serosa, what you're looking at here are extensions of what are known as the parietal peritoneum. And so just a quick review here for tissues and layering. You're looking inside of the abdominal cavity, and all these digestive organs we're talking about more or less hover in the middle of the digestive abdominal cavity. On the outer or the innermost surface here of the wall of the abdominal cavity, you have what you can think of as wallpaper covering the walls. This is known as the parietal peritoneum. Now, what happens here is the parietal peritoneum does cover the surfaces of these walls, but at certain places it can extend away from the wall and actually reach out to some of the organs in the cavity. Now, as it wraps around these individual organs, we rename it. Now it's still called peritoneum, but now it's called the visceral peritoneum. So take a look at a few examples. So here you see parietal peritoneum on the wall, and then it extends to the liver and covers it in visceral peritoneum. Here you can see again parietal peritoneum on the wall, but around the stomach right here, it's visceral peritoneum. Same thing here with regions of the colon and the small intestine. Now you do have plenty of organs that are back behind all of this. So you can see the pancreas, right? Parts of the small intestine, parts of the large intestine, the urinary bladder, right here the rectum and the sigmoid colon. Those are what we know as retroperitoneal because they're behind the parietal peritoneum. They would not have that outermost tunic that we're talking about, but they're in the minority. There's a few regions here that have specific names beyond visceral or parietal peritoneum, and those are known as mesenteries. So a mesentery itself is just an extension of this parietal peritoneum, but it's adapted to hold on to things like fat, lymph nodes, lots of vasculature, and so it actually in recent years has become known as an organ in its own right because of the many different tissue types it holds. And so the ones you can see here, the lesser omentum running from the liver to the stomach, the greater omentum running from the stomach down and covering the face here of your small and your large intestine, and the mesocolon. And the mesocolon itself is going to be anchoring parts of the colon, right, also known as the large intestine, right through here. I think this image does a nice job of showing each of them: lesser omentum, greater omentum, and here the mesocolon itself. Make sure you're familiar with the differences in the terminology here, the types of peritoneum, and of course what the mesentery types are, so that you can identify what they are through an explanation from me. So of course with digestive processes, we're going to spend most of this PowerPoint just taking a look here at whether organs are classified as alimentary canal or accessory organ. And in Chapter 25B notes, we'll look more specifically at the different functions that they are capable of. And so I'll go ahead and give you a brief rundown of what each function is. Ingestion is selective intake of food. As you can see, that's only done here in the mouth or the oral cavity. Propulsion is movement of food through the alimentary canal. There's two ways this can happen. That's through peristalsis or segmentation. And at a quick glance, you see that most alimentary canal organs are going to facilitate propulsion. Now let me break away to the next slide before I come back. Peristalsis itself is waves of contraction that are meant primarily for propulsion. Now, segmentation itself is still going to move food forward a little bit, but its primary function here is to mix food together. And so what you're seeing in this image is three separate sections of boluses, so that's food pellets essentially. And as smooth muscle in the wall begins to change its rhythm of contraction, it begins to mix each bolus with the one it's adjacent to until all of them are mixed equally. Okay, so again, segmentation itself, while it helps with propulsion, does tend to be most associated with the next process, mechanical digestion. So mechanical digestion is a physical process. It mixes food, it breaks down large foods. As you'll see here, that this mechanical digestion can be carried out in the mouth, right, with teeth and tongue. It can be carried out through the stomach with contraction itself. The small intestine right here is the last place we see mechanical digestion using segmentation. You'll also see chemical digestion. So this is digestion carried out by a series of hydrolysis reactions. Hydrolysis means to cleave large molecules into smaller molecules. And so you'll see here that salivary glands, the stomach, liver, gallbladder, pancreas, the small intestine, those are all capable of facilitating chemical digestion. Now, once nutrients have been digested and broken down, you are free to absorb them. And this is done at specific places. You'll see that the majority of absorption is carried out here through the small intestine, while some absorption, mostly just that of water, is carried out by the large intestine. Compaction helps you to consolidate indigestible residue into feces. That's going to be carried out by the colon or the large intestine. And of course, defecation itself only carried out here by the rectum and the anus. So make sure you know which digestive processes are carried out by each organ. And of course, understand how all of this is going to be regulated. And so there's 3 primary forms of regulation of all these digestive processes. The first involves the nervous system. And so the first and the second involve the nervous system. And so we see first here what are known as short or myenteric reflexes. And this is what we know of as intrinsic regulation. So this is what's happening within the organ results in a reflex that changes that organ's function. So when we take a look here at this particular image, a great example of a short reflex is seen right through here. So if this is going to be the wall of a hollow organ, pretend it's the small intestine, here is its lumen, then essentially what's happening here is anytime you have changes in the chemical content or the stretch of this organ as food enters it, then a short reflex can basically trigger effector muscles like smooth muscles or glands to change contractile or secretory activity. Now this is going to rely on the nerve nets that you see within this organ itself, and it doesn't rely on any kind of input from higher level parts of the nervous system like the brain. So it's a short reflex. By nature, it's fast. When you take a look here at long reflexes, these are called vagovagal because most of them are mediated through the vagus nerve. And so here, when you see these vagovagal long reflexes, they're long because they take a long signal pathway, a long nerve coming from the brain, and they relay information from the brain all the way down to that organ. Of course, anytime you have motor function coming from the CNS down to one of these target organs, you typically also have sensory function through visceral afferents. And so any kind of chemical, osmotic, or mechanical change in that organ is going to be transmitted here to the CNS, which then is going to send output, motor, and change here the effects of the smooth muscle or the glands. So it's long reflexes because of long nerves, but by consequence, it does also sometimes take a little bit longer to start. And finally, you have chemical regulation without the use of neurons, and this is done through enteroendocrine cells. And so these are cells that are going to be embedded in the mucosal layer, and they can release a widespread number of paracrines and hormones to regulate the function of either the digestive organ itself or nearby digestive organs. And so we'll see several of those as we move through. And so in lecture, what we'll do is we'll go through some of these questions about the notes. So the first one is this: the myenteric nerve plexus that is associated with short reflexes is a part of which digestive organ tunic? And my apologies, I've corrected here. This should say with long reflexes. So the myenteric nerve plexus that is associated with long reflexes is a part of which digestive organ tunic? A, the mucosa, B, the submucosa, C, the muscularis, or muscularis externa is what I'm looking at there, D, the serosa, or E, the adventitia. So take a minute, see if you can answer it, and then move to the next question. Question 2. Which of the following organs is considered part of the alimentary canal? Is it A, the stomach, B, a salivary gland, C, the liver, D, the pancreas, or E, the gallbladder? In the next video, we're going to start looking at specific organs running from the mouth to the stomach. So take a minute, recap, get that lecture ready to go, and then watch the next video.